we've been looking at how one characteristic is inherited from parents to offspring. And we've been looking at an example of flower color. So Mendel, this is Mendelian genetics because this is a study that Mendel did looking at garden peas. So when he took true breeding garden peas and that have purple colored flowers and crossed them with a true breeding white flower color plant, he got 100% purple flowers. So what do I mean by true breeding? And how did he know that these are true breeding? So he bred them over and over, many generations, to make sure that there were no alleles in there. He didn't know that they were called alleles at the time, but that there were no latent factors, is what he called them, or now we call them alleles, that are recessive in there, that they're always dominant, that all of the plants, that these true breeding purple flower colored plants have all dominant alleles. And right, so he figured that out by breeding them over and over and over again. And so never did the recessive allele ever show up. So that's how he knew that they were true breeding, and that these factors, that it had both of the expressed factors, or now we call them dominant alleles, that this plant had both dominant alleles. Okay, so we know from studying meiosis that the gametes that this individual right here with a purple flower can produce, because, and let's go ahead and draw that out, these are our chromosomes from mom and from dad, and it has the allele on it for the protein that makes the pigment. So we know that in S phase, these are going to be replicated. And then we also know in meiosis that these are all going to separate into gametes. These sister chromatids are all going to separate. They're going to become daughter chromosomes in gametes. So we know that this is this strand of DNA is going to go in one gamete. This strand is going to go in another gamete. This strand is going to go in a third gamete, and this strand is going to go in a fourth gamete. And because it's true breeding, we know that this allele, all of these alleles are considered big A dominant. So when we do a Punnett square, and let's do that down here. We can do a Punnett square right here, that all of the gametes from this individual are all going to be big A. So these are all gametes. You can even draw a picture around them and say, okay, these are the sperm from the male. And then this individual right here, again, if it's true breeding white, then we know that all of these alleles are A, little a, little a, little a, little a, and you can say these are the eggs from mom. So these are the gametes from dad, and these are the gametes from mom. And you know that all of the individuals that these two parents can make are all going to be big A, little a, every single one of them. And that's why you get 100% purple flowers in the first generation, because all of the offspring are all going to be big A, little a. Okay, so what about the second generation? So we've looked at that, and we know that so an individual can have 4 gametes, and 2, when you cross the first generation with self-fertilization, you know you're going to get 2 big A's and 2 little A's. So really we could have put in here, we could have put another row over here with big A's, and we could have put another row over here with little A's. But we're going to get the same ratio. So we can go ahead and just put big A and little a. And we know we're going to get the ratio of 3 to 1. So we're going to get the phenotype is going to be 3 to 1. So 3 purple flowers to 1 white flower. And our genotype, we are going to have 1 big A, 2 big A, and then 1 little a. And so our big A is called homozygous dominant. Our little a, since these are the same alleles, they're going to be homozygous recessive. And then big A, that's going to be heterozygous. Okay, so let's take a look at what happens looking at a different characteristic. So let's say, let's look at seed color. So our phenotype for seed color is yellow seed or green seed. And in the first generation, we have 100% yellow. So we know that yellow is dominant. In the second generation, if we were to self-fertilize the plants from each of these seeds, we would get 75% yellow and 1% green. So 3 to 1. And so Mendel produced many, not just 4, didn't just grow 4 plants from this. He grew hundreds. So he had very large numbers to show that this wasn't just by chance, and that this was a real pattern that happened for every characteristic. So these are our phenotypes. Our genotype, we are using letters. So big capital letters for the dominant allele and lowercase letters for the recessive alleles. So our first generation, we are going to have a heterozygous individual. So this is heterozygous. And that means that there are 2 different alleles. And in our second generation, we are going to get a homozygous dominant, and a homozygous recessive, and we are going to get heterozygous. So these, remember, these represent the alleles in the genome. Genome, let's write genome. What do we mean by genome? Genome is all DNA. All of the DNA in an organism this is protein coding and non-protein coding. Okay, So let's take a look at what happens if you have two characteristics. Actually, let me go back and wait a minute. Let me go back. So I wanted to define one more thing. So monohybrid cross. That means that we're looking at just one characteristic. So we're just looking, in this instance, we are just looking at seed color. So here is our homozygous dominant. It's a true breeding. And here is a homozygous recessive, also true breeding. And all of the gametes that this individual can produce are all going to have a recessive allele. And in this case, we're going to represent it with a lowercase y. All of these individuals are going to have uppercase y gametes, so all dominant alleles. When these gametes come together in fertilization, we're going to get our heterozygous F1 generation, and all of the offspring are going to be heterozygous. That's the genotype, and they're all going to be yellow seed. That's the phenotype. Okay, so now what gametes can the F1 generation come up with? They can have dominant alleles, and they can have gametes and gametes with recessive alleles. Okay, so we are going to put those into a Punnett square. So in this case, we're going to have, this is going to be the gametes that father can make, and let's say that these are the sperm, and then these are the gametes the mom can produce, and these are the eggs. And these are all of the offspring that these two can produce. So they can produce 3 yellow-seeded plants to every 1 green-seeded. So a plant with green seeds. So yellow, 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 green, that's the phenotype, and the letters represent the genotype or the alleles that are represented in the genome. And then we've got the ratios down here. Okay, so that's a review of one characteristic. So next we're going to take a look at a dihybrid cross. So when we're looking at two characteristics, how do we put two characteristics into a Punnett square? Okay,